You are what your ancestors ate. It's an old saying, but a newly published genetic study proves it's literally true. Like, literally.
Yeah, it really is. And, welcome to the deep dive. Usually when we think of human evolution, we picture it as this incredibly slow, ancient process, right?
Yeah.
Like it finished a long time ago.
Right, like we treat the human genome like it's a static blueprint locked away in some museum display case.
Exactly. But today, we're going to look at your DNA as a living, editable document. Because if your ancestors ate enough of a certain food, your body actually copy-pasted specific genes over and over again just to handle the load.
It's just wild. And for you listening, our mission today is to unpack this dense, genuinely fascinating genetic research paper from Nature Communications. It was published in May 2026.
Yeah, and the goal is to break down every mechanism, every detail, and the algorithms they use to understand how a specific diet, specifically potatoes, rewrote the DNA of an entire human population, the Indigenous Peruvian Andeans.
So to really grasp the magnitude of this, I feel like we first need to understand the gene at the center of the story.
Right, so it's called the AMY1 gene.
Yeah.
AMY1. And this gene produces an enzyme called salivary amylase.
Okay, salivary amylase.
Yeah, so if you've ever chewed on a piece of bread and noticed it getting slightly sweet after a minute or two.
Oh, yeah, like a saltine cracker or something.
Exactly. That is salivary amylase at work in real time. It is the enzyme in your saliva that starts breaking down complex starches into simple sugars the absolute second that food enters your mouth.
Right. So it's the very first step in carbohydrate digestion.
Right.
But let me make sure I have this straight. Let's unpack this a bit because we all have this gene, obviously. We all digest carbs.
Right.
But usually I think about genetics as getting one copy of a gene from your mom, one copy from your dad, and, that's the end of the story. What does it mean to have copies of a gene? How does a population just get more of them?
That is the standard assumption, yeah. But this gene is a wild exception. Human biology doesn't restrict itself to just 2 copies of amylase. The scientific term for this is copy number variation, or CNV.
Copy number variation, okay.
Think of it like this. Imagine your genome is a massive recipe book. Most recipes are written in there exactly once. But for amylase, human genomes act like a broken photocopier.
A broken photocopier. I love that.
Right. Because globally, humans have anywhere between 2 and 20 copies of this exact same AMY1 gene hidden inside their cells per diploid genome. The global median is about 7.
Okay, so the global median is 7. But in this case study, the researchers analyzed an incredible data set, right? Something like 3,700 individuals from 85 different populations.
Exactly. 3,723 individuals to be precise. They found a massive outlier. Peruvians living in Lima, which is designated as PAL in this study, and the Acamel odom, also known as the Pima of North America, they have the highest median copy numbers globally.
Which is how many?
A median of 10.
Wait, I have to push back here. 10 copies. When you're dealing with massive genomic data sets and millions of lines of code, how do we know this isn't just a glitch in the sequencing, like a typo in the computer?
That is the exact skepticism you need. To prove it wasn't a glitch, they used a dual algorithm approach. First, they used a tool called mRCanavar. MRCANAVR.
Okay, what does that do?
It's a rapid read-depth-based caller. Basically, it maps short genetic reads against the standard human reference genome, which is called GRCH38. It looks for a massive pileup of identical genetic material in the AMY1 region.
But salivary amylase looks a lot like the amylase produced in the pancreas, right? Wouldn't a rapid scanner just get confused by that?
Precisely. The pancreatic cousin is called AMY2. So they paired mRCanavar with a second algorithm called genitocien. This one is highly meticulous. It acts like an intense proofreader to perfectly distinguish between the AMY1 gene and the AMY2 gene.
Oh, wow. So they double-checked the computer's work.
And they didn't stop there. They took these computational counts into a physical lab and validated them using ddPCR.
Digital droplet PCR, right?
Exactly. It's an advanced physical counting method.
Yeah.
And the results were stunning. They achieved extraordinarily high confidence R-squared values of 0.94 and 0.98. The mathematical alignment between the computer and the physical reality was near perfect.
So the 10 copies are definitely real, but, we know there's European and African admixture in South American populations. Could this high copy number just be leftover DNA imported from somewhere else?
They ruled that out brilliantly. They set up a control comparison. They looked at indigenous Quechua from Peru who live at high altitude and have that massive median of 10 copies.
Right.
And they compared them against the indigenous Maya from Mexico, who live at a low altitude and have a low AMY1 median of just 6. Even when they strictly filtered the data to look only at segments of DNA with purely indigenous ancestry, the Quechua's massively high copies remained.
So it's an authentic, highly localized indigenous trait.
Exactly.
Okay, before we go deeper, let me just jump in for a quick recap. Let's bullet out what we've learned in our first block here.
Sounds good.
So, bullet 1. AMY1 digests starch, and humans vary wildly in how many copies they have. Bullet 2. Indigenous Andeans have the highest global median at 10 copies. And bullet 3. Rigorous algorithms like Amercanavar, Genotocien, and ddPCR prove this isn't a sequencing error or some modern genetic mix-up. It is real.
Spot on.
And for you listening, think about your own diet. If your DNA could make extra copies of an enzyme for the food you eat most, what gene would you have 10 copies of? It's pretty crazy to think about.
It really is.
But so moving on, they have more copies. But couldn't that just be random chance, like a roll of the genetic dice when they migrated to the Andes?
That's the big question. You're talking about genetic drift versus positive selection. When populations crossed the Bering Strait and migrated to South America, they faced extreme population bottlenecks.
Which causes genetic drift, right, where small groups splinter off and dumb luck dictates what traits survive.
Exactly. So to prove this is selection meaning survival of the fittest, and not just dumb luck, the researchers brought out the heavy statistical artillery. They looked at FST and VST. VST measures the variance in copy number, and FST measures the variance in single nucleotide variants, or SNVs.
Okay, let's zoom in on that. Single nucleotide variants. Those are like single-letter spelling changes in the DNA.
Right. Think of them as genetic flags or markers. And AMY1 ranked in the top 0.07% of all genome-wide variations between the Quechua and the Maya.
Wow, top 0.07%. So it wasn't subtle.
Not at all. They found specific tag SNVs. The first one is RS586777276. It's associated with low copy numbers.
Okay, and who has that one?
It's found in about 26% of the Maya population, but it is completely absent 0% in the Quechua.
Wait, literally 0%. Evolution just wiped it out.
Yeah. And conversely, the star player is a flag called RS143597860. This one flags high copy numbers. It's present in nearly 46% of the Quechua, but only about 14% of the Maya.
But again, to play devil's advocate here, how do we know that jump to 46% wasn't just genetic drift from that bottleneck we talked about?
Because of the neutral simulations. They use an algorithm called EMS prime, MSPRIME.
And what does EMS prime do?
It literally simulates human history. They simulated 3 billion base pairs of DNA under different historical population sizes to see if random chance could ever make that high copy flag jump to 45%.
And what did the simulation say?
It was mathematically impossible without positive selection. They even dropped the simulated Quechua population size to an unrealistically tiny group of just 250 people, desperately trying to force a scenario where genetic drift could explain it.
Oh, to try and artificially create a super tight bottleneck.
Exactly. Yeah. And it still couldn't happen by chance. Plus, they ran advanced selection tests like PBS population branch statistic and Ohana, which account for admixture using Finnish, Spanish, and Yoruba outgroups. The high copy SNV still ranked in the top 0.01% for selection.
That is just incredible math. But didn't they also look at the physical structure of the DNA using XPEHH?
Yes, cross-population extended haplotype homozygosity.
Okay, that is a mouthful. Let me try an analogy here to see if I understand the mechanics of this.
Go for it.
Imagine a long train of boxcars where each boxcar is a piece of DNA. Usually, as generations pass, recombination acts like a train yard switch, right? It uncouples the cars, shuffles them around, mixes genes from both parents.
Right. Recombination breaks up the sequences over time.
But if a specific trait is incredibly beneficial for survival, evolution says, do not touch this. The whole untouched train of boxcars survives together across generations without getting shuffled.
That is an excellent way to picture it. And the XPEH algorithm specifically looks for those unbroken trains. The value for the Quechua was over 6. Which puts it well into the 99th percentile.
Over 6, that's huge.
It proves a massive unshuffled train of AMY1 copies was actively preserved. The evolutionary signal for this starch-digesting gene was actually stronger than the signal for EGLN1.
Wait, EGLN1, isn't that the famous high-altitude survival gene?
Exactly. The one that lets Andeans breathe in extreme, low-oxygen environments. Evolution prioritized digesting carbs even more fiercely than breathing thin air.
That's staggering. Okay, so subsummary time. Using FST, XPEHH, and computer simulations, researchers mathematically proved this wasn't an accident of history. It was actively selected for survival.
Correct.
And for the listeners, it is incredible to think that math can peer 10,000 years into the past. Does it change how you view your own biology? Knowing statistical algorithms can read your DNA like a historical receipt.
It really reframes things.
So this brings us to the next massive piece of the puzzle. Here is where it gets really interesting. If this was an evolutionary choice, when did it happen, and how did the DNA physically alter itself?
Let's tackle the timing first. They used an algorithm called RELATE, R-E-L-A-T-E. It builds ancestral recombination graphs to estimate how common these alleles were over time.
Like working backward through a family tree of mutations.
Basically. And they had a massive aha moment. The algorithm showed that exactly 10,000 years ago, the frequency of that high copy SNV, the RS143597860 flag, just skyrocketed. It went from a baseline of 2% up to 45%.
Wait, 10,000 years ago, isn't that exactly when agriculture started?
It is a perfect coincidence. It perfectly aligns with the domestication of the potato, roughly 10,000 to 6,000 years before present, in the Lake Titicaca region.
Oh, wow. Lake Titicaca.
Yeah, the ancestors of the Andeans settled there and radically shifted their diet. They began consuming heavily starch-based root vegetables. They had a selection coefficient of 0.0124 and a log likelihood of 11.15. The math perfectly matches the historical agricultural shift.
Okay, that paints an incredible historical picture. But there is a physical mechanism question here. How does a gene actually duplicate itself? Because the body doesn't just magically manifest new DNA out of thin air.
Right, it requires a mutational mechanism. And to solve this structural puzzle, the rapid short-read scanners weren't enough anymore. They had to use Oxford nanopore long-read sequencing.
Because the short scanner only looks at tiny pieces.
Exactly. If a gene repeats 10 times, the short reads all look identical. You can't see the larger structure. But long-read sequencing reads massive, continuous chunks of DNA. They achieved an N50 of around 20 kilobases.
So they were reading 20,000 base pair chunks at a time.
Yep. It's like having puzzle pieces that are whole paragraphs instead of single letters. This allowed them to rule out a mechanism called MMBIR, microhomology-mediated break-induced replication.
How did they rule that out?
Because the AMY2 genes weren't duplicated along with AMY1. It was very specific. The actual culprit is something called NAHR, non-allelic homologous recombination.
Okay, non-allelic homologous recombination. Let me try another analogy for this one.
All right.
Imagine proofreading a book, right? And there are two identical paragraphs existing on different pages. You're reading, you blink, and you accidentally jump from the first page to the matching paragraph on the second page. Because they look so similar, you lose your place and accidentally copy-paste everything in between twice.
That is exactly what happens. The DNA sequences on either side are so similar, the biological machinery accidentally loops back. The specific unit of repetition they found was an AMY1A and an inverted AMY1B sequence.
And long-read sequencing actually let them see that loop.
Yes. In fact, here is a fascinating detail. When they analyzed ultra-long reads of a modern 14-copy Peruvian genome, so 7 copies on each chromosome, they found the best genetic match was actually a known 9-copy Andean sequence, just missing one 2-copy mutational unit.
Wait, meaning what?
Meaning the high-copy sequences weren't imported from outside. They originated locally in the Andes from existing variations.
Yeah.
Evolution just kept taking advantage of this NAHR copy-paste error because it was so incredibly useful.
That is wild. Okay, let's pause for our second milestone recap here. Let's summarize the middle of our deep dive.
Let's do it.
Bullet 1, statistical algorithms like XPEHH and EMS' proved this was intense positive selection, not random drift. Bullet 2, the timing 10,000 years ago precisely matches the domestication of the potato.
Right.
And bullet 3, the DNA physically expanded through a biological copy-paste error called NAHR, which evolution kept because eating potatoes was suddenly crucial for survival.
Perfect summary.
So for the listeners, think about this. What cultural habits do we have today that might be creating evolutionary pressures for humans 10,000 years from now?
It's a sobering thought.
It really is. So what does this all mean for the person walking around with 14 copies of AMY1 today? Like the phenotypic so what? Do they just digest potatoes like a superhero?
Well, yes and no. Biology always demands a trade-off. Let's look at the downstream effects. Having more gene copies means more enzyme dosage. You have a massive volume of amylase in your saliva.
Right.
In the gut, high MY1 is strongly associated with gut bacteria that can digest resistant starch. This optimizes energy extraction. You literally get more calories out of a potato. In fact, they noticed another gene, the MGM gene, which is a downstream maltase processor, evolved simultaneously.
So the entire starch digestion pathway adapted together in the gut. That sounds like a superpower. Where's the downside?
The downside is in the mouth. Higher AMY1 levels are strongly linked to pathogenic oral bacteria, specifically Streptococcus.
Oh, because there's so much sugar being rapidly broken down right there in the mouth.
Exactly. It feeds cavity-causing bacteria before the food even reaches your stomach. The evolutionary trade-off for extracting maximum energy is terrible dental health. In Peru today, over 85% of school-aged children report having cavities.
85%. That is a massive phenotypic cost.
It is. But when the alternative is starvation, cavities are a survival compromise. And it brings us back to the Acamel odom, the Pima population in North America. Remember, they also have a median of 10 copies.
Right, because they adapted to corn, which is another heavy starch.
Exactly. They developed high AMY1 to survive a maize-based agricultural diet. But today, the Pima suffer from incredibly high rates of metabolic disease.
Wait, if their bodies are optimized to process starch, why are they suffering from metabolic disease now?
Because a genetic advantage for an ancient, whole food agricultural diet offers no inherent protection against modern, highly processed diets. In fact, if you introduce synthetic processed carbs to a body optimized for squeezing every calorie out of a raw starch, the machinery goes into overdrive.
So it's a massive biological mismatch. So just to summarize this final point, extra AMY1 optimized energy extraction from potatoes, but the trade-off was poorer oral health and an increased vulnerability to modern metabolic diseases.
That's the reality of evolution.
And for you listening, evolution is always about trade-offs. Would you accept a higher risk of cavities if it meant your body could extract more pure energy from the food you grew?
It's quite the dilemma.
It has been an unbelievable journey today. I mean, we've gone from microscopic genetic copy-paste errors like NAHR and algorithmic data from mCAR-Canavar, FST, and XPEHH, all the way to the grand 10,000-year-old story of Andean farmers domesticating the potato.
And as we wrap up, I want to leave everyone with a final puzzle to mull over. We've seen how our DNA rapidly adapts to the food we grow. But today, our diets are changing faster than our genes can mutate. If it takes 10,000 years to adapt to the potato, how is our ancient AMY1 machinery going to cope with the ultra-processed synthetic carbohydrates of the 21st century?
Oh, man.
Are we currently living through a biological mismatch that we won't even understand for another 10 millennia?
That is profound. It makes you look at a bag of potato chips completely differently. Thank you for joining us on this exploration. Keep asking questions about the hidden history written right there in your own cells. See you next time.